Good evening, viewer. Welcome to our FTV English version. Viewers, the human genome consists of 3 billion base pairs, and when a cell divides, it takes about 7 hours to complete making a copy of its DNA. So that's almost 1,20,000 base pairs per second. At that breakneck speed, one might expect errors to occur, and they do at a rate of about 2% per second in every dividing cell. But cells have a DNA repair kit too. They are enzymes that can correct those errors at a rate matching that at which they occur. A bigger problem happens when there is a barrier to DNA replications, the process of copying the DNA. It can lead to break in the chromosome, which will lead to loss of vital genetic information if not repaired. Gaps or breaks in DNA can be potentially harmful or fatal should they lead to genetic diseases or cancer. Catherine, professor of biology, has been examining cell DNA repair response to these critical events in yeast cells as an analog to human cells, and has discovered that the process is more elaborate and layered than previously thought. In a recent study published in Cell Reports, scientist and his co-authors Tyler Macaulay, Jenna Wallen and Matthew Johnson looked at areas of the DNA that are particularly susceptible to breakage. Those areas are where the sequence consists of long stretches of repeated triplets like CA, GCA, GCA, CAG, or couplets like ATA, TA, TAT, which continue from just a few to hundreds of units long. When that occurs, the DNA may not always fold neatly into a long double helix, but may twist on itself to form hairpins and cruciform structures like a tangled electric cord. So this is not a minor issue because repetitive DNA makes up about 10% of our genome, which is even greater than the portion that codes for protein, says scientist. Behind this exclusive report,